Have you ever wondered how the reactions taught in your organic chemistry class are used to create molecules that treat disease and save lives, while generating millions for pharmaceutical companies each year? In this series, I'll be dissecting the top 200 pharmaceutical sales for 2022, discussing the syntheses of each molecule and how they treat diseases. Deferasirox, sold under the brand name Exjade, was the 200th top-selling pharmaceutical in 2022. It was developed by Novartis and approved by the FDA in 2005. Deferasirox is used to treat chronic iron overload caused by long-term blood transfusions for conditions such as beta thalassemia. It works by chelating excess iron in the blood, allowing it to be excreted from the body. Starting with a retrosynthesis of Deferasirox, it is important to know that when you see a heavily substituted 5-membered heterocycle, it is most likely formed through a ring synthesis rather than functionalization of a core. Furthermore, when disconnecting ring systems where two heteroatoms are bonded together, these bonds are normally introduced rather than formed. In this case, the triazole could be formed from a phenylhydrazine and an imide group, which could be further disconnected to a carboxylic acid and an amide. In the forward synthesis of Deferasirox, this carboxylic acid and amide were refluxed with phenyl chloride to form the imide bond, which underwent an intramolecular cyclization to give the masked imide intermediate. Reaction with phenylhydrazine resulted in formation of the triazole ring, giving the final product. Next on this list is Orenitriam, a brand name for an orally available form of the drug Tresprostinil, which can also be administered by infusion or inhalation using different formulations. Tresprostinil is used for the treatment of pulmonary arterial hypertension, a condition characterized by high blood pressure in the arteries of the lungs. The compound is a vasodilator, relaxing the blood vessels and allowing blood to flow more easily, which reduces the workload on the heart. Tresprostinil is a synthetic analogue of prostacyclin, a naturally occurring prostaglandin. The development of Tresprostinil was prompted by the need for a more stable and long-acting alternative to prostacyclin, which has a very short half-life and requires continuous infusion into the blood. Synthesis was initiated by the protection of the benzyl alcohol with TB-DMS chloride. This was then subjected to a direct methylation using butane and ethyl vinyl bromide as an electrophile. The alcohol was then deprotected using T-bath and oxidized to the aldehyde using a Swern oxidation, creating this alkene fragment. The second fragment started with an epoxide ring opening on a chiral epichlorohydrin building block using butyl magnesium chloride. This gave a secondary alcohol that could be protected as a THP group. A lithium alkyne species was then used to displace the chloride leaving group, with the TMS group on the alkyne being removed using sodium hydroxide. This fragment was then reacted with a Grignard reagent to undergo metal-hydrogen exchange on the terminal position of the alkyne, which subsequently attacked the aldehyde group, forming an alcohol in the process. This will give a mixture of diastereoisomers, as the addition of the alkyne to the aldehyde will have no stereochemical preference. The alcohol was oxidized back to the ketone, and then a CBS reduction was used to install the correct stereochemistry. The alcohol was then protected to allow for the key reaction to take place, Parkinson-Calm reaction, which forms two of the rings in one step. A series of reductions then took place, the first using hydrogen and palladium over charcoal, and then a second using sodium borohydride. PTSA was used to deprotect the THP group, and lithium diphenylphosphide was used to demethylate the aromatic methoxy group. The phenol could then be alkylated with chloroacetonitrile, which could be hydrolyzed to reveal the carboxylic acid group, completing the synthesis. At 198 is Rotigotine, sold under the brand name Neupro. It is a non-selective dopamine agonist and is used to treat Parkinson's disease and restless leg syndrome. It is administered via a transdermal patch, allowing a slow and constant release of the drug. When thinking about a retrosynthesis of Rotigotine, you should notice the tri-substituted amine in its structure. Reductive amination is a key reaction used to form carbon-nitrogen bonds and can be used to form two of these bonds, leaving us with propylamine as a nitrogen source. To kick off the synthesis, a monomethylation of the following dihydroxynaphthalene was carried out using dimethyl sulfate. The ring system was then partially reduced by a Birch-type reduction using sodium metal dissolved in ethanol. The first reductive amination could then take place using propylamine and hydrogen with a platinum catalyst as a reducing agent. The racemic mixture could then be resolved using dibenzyl L-tartaric acid, and the methoxy group was demethylated using HBr. The final step is reductive amination, where the aldehyde is formed in situ from its corresponding carboxylic acid using borane. The imine formed through the condensation of these two groups can also be reduced by borane, giving the final product. That is all for this video. Thank you for watching. If you want to see more videos like this, be sure to like the video as it's the best way you can help support the channel. References to papers used will be in the description, and if you have any questions, please leave them in the comments below. Also, I'll be doing a giveaway once we reach 1,000 subscribers, so stay tuned if you want to hear more about that.